basically, for example, there's a method to evaluate the oligonucleotide, the distribution of that, and to see if they have certain pattern, and they just use this pattern to construct a distance-based phylogeny, and the result is not too bad actually. So this is the ribosomal RNA tree, and this is the new approach to identify a certain oligonucleotide, the presence or absence. And the two trees are highly identical, mostly. Okay. And the problem, of course, is we have no idea about the mode of evolution. How do those oligopeptide or oligonucleotide actually the meaning of those oligonucleotides? We have very little idea. So, but people still do that. And gene order information can also be added, because genome organization is very important information. So if different organisms have very similar gene order, they should be they should be closer in relationship. So people also put that into account, and then reconstruct the phylogeny only according to the gene order, and still can be reconstructed solely by the gene order, and still get a very similar phylogenetic information compared to the sequence-based phylogeny on the right. So it's also basically very compatible, and we have no idea if we can combine those two methods, or if it's good to combine it. Okay, and the gene content method is to create a new data matrix to press the presence and absence of a specific genes. So anyway, this is one way to do it, and it's doable, and the result again is promising. But not that many people using it anymore, because it's very easy to get sequence data nowadays. There are some programs available just for your information, and so in summary, those supertree information or supermatrix reconstruction is to get the data and separate those into supermatrix or supertree approach and get a tree. And sometimes you can use the whole genome features to do that as well. So this is the summary for the lecture so far. And here are some of the new approaches we nowadays get, is to get quite a lot of ESD data from NGS data, and we'll have very easy just to spend some money, and you get a lot of sequences. Then how are we going to deal with it? And basically these are the pipelines, and we got NGS data, and those can be assembled to the unigenes, and then we perform a broad search, and to only accept one gene for one ortholog for one taxa, and for that purpose you can get a reliable alignment according to it. And if not, we can just go to try to figure out what is the real ortholog of the gene. If we cannot, we could just basically eliminate the data. And we eventually got a alignment and make a tree accordingly.